Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon Incorporated, I'd like to welcome you to Overcoming the Microbiome Challenge, a panel discussion. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now, I'd like to introduce our moderator and first speaker for today. He is Gregory Michelotti, PhD, Scientific Director for Metabolon Incorporated. Welcome, Dr. Michelotti. The presenter ball is yours.
Thank you very much, and welcome to everyone to our microbiome panel discussion, where we will hear how metabolomics is being used to overcome challenges in microbiome research. Today, we're going to hear from two key opinion leaders in the field who are leveraging the power of metabolomics to understand the role of the microbiome in human health. In the first talk, Nikole from Siolta Therapeutics, and I'm thrilled that I think I came close to the Gaelic pronunciation on that, is going to describe her company's approach in using biochemical profiling to understand the role of microbial communities in chronic inflammatory diseases, such as asthma that you'll hear about today. And this will help identify new treatments and stratify populations for the most effective and personalized treatments. And in the second talk, Eran Segal from the Weizmann Institute in Israel will discuss his laboratory's recent advances in combining metabolomics with machine learning approaches to help elucidate the role of microbial activity in mediating differential glycemic responses to diet in humans. The ability to accurately predict personalized dietary responses has enormous implications for successfully and naturally normalizing glucose levels in individuals predisposed to diabetes. So we are thrilled to have such preeminent investigators here to participate with us today. So this is the overall agenda of today's discussion. I'm going to provide a brief overview, showing the value of metabolomics for studying the microbiome, and then we'll have our two talks, the first from Nikole and the second from Eran, and then a Q&A session afterwards after both talks. Before we get to the science, however, I'd like to provide a brief overview of Metabolon, who we are. We have been generating metabolomic data for the research community for over 20 years. And because of this extensive experience, and you can see all the parameters of which we consider our success on the slide here, from a technical standpoint, we've been able to build out and refine our platform to provide the most comprehensive coverage across all biochemical classes while maintaining the lowest CVs or variability in the field. Now, this is particularly exciting for us since there has been an acceleration in the use of both targeted and untargeted biochemical profiling, and metabolomics is increasingly being recognized as a central modality in biochemical research. Within this context, metabolomics is unique in that it integrates both genetic and non-genetic influence to provide a comprehensive view of health status or a biological system. On the top, I think most of you will recognize the central dogma of molecular biology, the flow of information from genes to RNA to proteins. And then down below, you'll see that there's a non-genetic constellation of factors that are also incorporated into metabolomics profiling that are not traditionally genetically informed. And these include diet and xenobiotics, the microbiome, which we'll hear about today, and lifestyle and associated risks with that. And what I'd like to point out is that there is a level of complexity on the top, the genetic information, that while genes are very powerful and obviously a great deal of information and technology has been invested in this right now to understand the role of genes in human health and wellness, there's been a relatively limited impact in terms of how this information has resulted in therapies and insight into diseases. And there's an enormous level of complexity and understanding this risk because genes and RNA and protein are subject to a bewildering array of modifications and regulatory factors that confound our ability to understand how they operate in different processes, in different model systems, and even within different individuals. And these modifications include editing and splicing and post-translational modifications and sequestration that really confound our ability to understand how these factors behave in different model systems. Whereas metabolomics is unique that it integrates both this genetic and non-genetic influences to provide a comprehensive view of health status. And they are unique, biochemicals are, in that they are conserved across the phylogenetic spectrum so that the biochemicals, the small biochemical classes that we quantify, are really conserved from bacteria to humans so that succinate or histidine are structurally identical across the phylogenetic spectrum. And that structural conservation implies functional conservation, providing a level of translatability across all model systems. so that together metabolomics integrating this information really provides a definitive representation of the phenotype. And metabolomics is uniquely poised to help deconvolute the role of the microbiome, since biochemical messaging is really the language by which microbes communicate with their host. And this slide depicts the wide array of biological functions that microbes influence. You can just go through the slide, and it's everything across the spectrum of nutrition and immune responses and breakdown of food products and drugs. So it's no surprise that perturbations in microbial composition can lead to alterations in a phenotype. And I think that's depicted in this slide here, showing, at least in the context of the microbiome, all the reports, and each of these perturbations or disease states are supported by a large constellation of publications that support these. So that changes in the gut microbiome affect, of course, GI function, such as IBD, inflammatory bowel disease, and other GI dysfunction, such as liver disease, but also non-GI related functions such as cardiovascular disease, pulmonary disease, or inflammatory diseases such as asthma, that Nikole will talk more about today, oral diseases and disorders, skin disorders, and even neurological disorders such as behavior and depression. And it's really striking in that context that changes in a gut microbial community or gut metabolism are affecting circulatory metabolites that then influence neurological systems. And a lot of papers are going on to support this in terms of microbial activities regulating autism and epilepsy and amyotrophic lateral sclerosis, or ALS, for example. And as I said, there's a number of publications in the field regarding this. So with that overview of really introducing of how metabolomics is being used to look at the microbiome, I now turn it over to our experts, and Nikole will be presenting the first talk.
Thank you for the introduction. Again, I'm Nikole Kimes, CEO and co-founder of Siolta Therapeutics, which is a company that I co-founded in collaboration with my mentor, Dr. Susan Lynch, a professor of medicine at the University of California, San Francisco, and in collaboration with Samir Kaul, who is a founding partner at Khosla Ventures. Together, we have a pretty bold vision for the future of healthcare, and it's one in which we integrate elements of precision medicine with prevention together with microbial therapeutics to alleviate chronic diseases, and we're starting with asthma. To give you a general overview of our approach, we are developing rationally designed live biotherapeutic products, and more simply stated, that means that we use human data from clinical trials to help us design mixed bacterial species consortia that have synergistic functionality. In parallel, we're developing diagnostics and biomarkers for patient stratification, and together we can utilize both of these strategies for prevention of disease in neonates and infants that are at high risk for developing allergy and asthma, as well as for therapeutic applications in those with established disease. Now, as a spin-out from academia at UCSF, we are firmly rooted in the exceptional academic research coming out of our co-founder, Dr. Lynch's lab, particularly with her focus on the early life microbiome and the role that that plays in developing allergy and asthma later in life. We've been able to secure some of the preclinical concepts from this research through intellectual property and translate it into a clinical program. And we have just recently successfully completed our first in human phase 1B safety trial in allergic subjects down to the age of 2. Chronic disease, as I hope everybody recognizes here, is really one of the greatest threats to human health in the 21st century, and it is the leading cause of both death and disability, not only in the US, but throughout the world. It encompasses many different diseases, a number of which are associated with disruptions to the gut microbiome. That includes allergic diseases such as asthma, metabolic diseases such as diabetes and obesity, and as well neurodevelopmental diseases such as Parkinson and autism. And across chronic diseases, we're seeing an alarming increase in the incidence. For example, in asthma, which impacts over 330 million people worldwide, that's almost the entire population of the United States. It's increasing at an alarming rate of 50% per decade. and it disproportionately impacts our pediatric population, so it is currently the #1 serious chronic disease affecting our children. And there's no cure, and that means that we are spending billions of dollars, $50 billion in the U.S. per year alone, simply managing the symptoms of a disease, using standard of care and even newer biologics that have variable efficacy and serious potential side effects. How does the microbiome play into this? And over the last decade, we've seen the accumulation of substantial evidence showing that the microbiome is essential to human health. As Greg pointed out, it can impact many different aspects of health. And I think it's an important note to recognize that the human microbiome harbors 100 times more genes than our human genome, which is important because this represents a vast functional repertoire, which can then impact things such as behavior, development, metabolism, and even immune tolerance. We also know that the gut microbiome is established very early in life by key founding species, which then dictate the environment in which subsequent bacterial species will accumulate and increase diversity over the first years of life. And this happens in parallel with immune maturation. And it's now well established that this process in early life is important in establishing and maintaining immune tolerance throughout life. And this led our co-founder, Dr. Susan Lynch, to hypothesize that if there is an alteration to this essential founding microbiome, would that actually play a role in disease development later in life? And in fact, there have been numerous epidemiological studies that help establish that early life exposures to things that perturbate the gut microbiome have an impact and are associated with the increased risk for allergy and asthma later in life. This includes antibiotic use, cesarean section, formula feeding. These are all associated with increased risks of allergy and asthma. So this brings us to the last decade of work out of Dr. Lynch's lab, which I don't have time to go into all of the details here. I do encourage you to look up the publications, particularly the ones presented here, if you're looking for a blueprint of how to really begin to put together a mechanism of action moving from the microbiome to an immunological outcome. And she started here with collaborators being able to show that the early life microbiota can be used to predict disease. Furthermore, they were able to show that these differential compositions in the early gut microbiome influenced metabolism and reprogrammed metabolomic impact, and that this has an impact on immune development. And in fact, the infants that are at high risk for developing allergy and asthma show immunological phenotypes that are associated with the later disease development. They went on further to use more targeted approach in which they found specific microbial genes in their products that were able to impact the immune response and act as biomarkers for this process. And moreover, in another study, able to show that there is plasticity in this early life microbiome and that it could potentially provide a novel mechanism for intervening in disease early in life before symptoms are occurring. And I just want to point out here that this has been validated in multiple infant cohorts. It's also, there's been similar results in independent laboratories. Brett Finley's work out of Canada, Hans Bisgaard out of Copenhagen. So similar results in that the early microbiome can be used to define at-risk infants. So with this, what Sue's work was able to establish was that those infants at higher risk for developing allergy and asthma have differences in their microbial composition, both from a phylogenetic and a functional perspective. More importantly, with the topic of today's discussion is that using untargeted mass spectrometry here at Metabolon, they were able to show that there's metabolic reprogramming. And this occurs generally in categories such as a lipid metabolism. But there's also ways to predict the metabolites that showed the most differentiation between the cohorts to then move into more targeted responses and showed that there's specific lipids, and in this case, 12-13-dihome is one of those of the oxylipins, which indicate that they are biomarkers for risk. These metabolic reprogramming and shifts were also then shown to have immunological impact in some pretty novel ex vivo assays in which the fecal milieu from the sterile or the cell-free fecal water was able to stimulate an immune response that again replicated the phenotype that you would associate with allergy and asthma. And indeed, these kids were 2 to 3 times more likely to develop allergy at 2 years of age and asthma at 4 years of age. And it's really all of this work that was the foundation of Siolta's platform for which we're developing rationally designed live biotherapeutic products in a data-driven manner that's human-centric. So let me spend a minute on what that means. We start with human data, and we use that data from clinical trials to help us rationally design specific mixtures of bacterial species that provide a synergistic functionality that is depleted in high-risk infants. And with those combinations, we can take it back into the lab and do preclinical optimization to identify our most promising lead candidates, which we then optimize for manufacturing and production, which allows us to move back into human clinical trials to establish efficacy. And in normal drug development, this would usually be a linear line, and you would think about moving the efficacious product to the commercial market, which we absolutely intend to do. But what we want to show here is that there's the potential to do a lot more. And by using these different analytical methods, metabolomics being a predominant one, we can actually start to tease apart responders and non-responders, which allows us to feed the data back into our platform and iterate to actually move towards patient stratification and more targeted therapeutics for a subset of the population. And this, again, is moving us towards precision medicine in the chronic disease space. Since this is a talk on overcoming the hurdles, I thought I would end with just a bit of a thought process on the innovation that we believe is required within clinical drug development. And traditionally, if you look on the left, classical drug development is where you have a single molecule with a specific target and a given output. And it's very linear. We're learning not only through the microbiome, but especially within this space, that biological systems can benefit from a systems perspective. These are complex environments in which there's interdependencies and functional redundancy. And the development of microbiome-based therapeutics is uniquely suited to help us work through a systems perspective, which will allow the acknowledgement of multiple mechanisms of action and really drive our ability to address redundant functionality and move towards more precise targeted therapeutics. And with that, thank you very much for your participation today.
And I think now we will go directly to Eran's talk, where he's going to talk about the microbiome at the crossroads of health and disease. Eran?
Hi, everybody. And yeah, thanks, Greg, and thanks for Metabolon for hosting this webinar. So my lab is at the Weizmann Institute. My background is in computer science and math, and for the past 15 years or so, my lab is a combined experimental and computational group, and we've been heavily engaged in studying the microbiome and its role in health and disease, with the aim of integrating it with genetics, lifestyle, nutrition, and try to arrive at personalized medicine approaches and precision medicine approaches tailored to people's unique composition of all of these different elements. In this talk, I'd like to cover one specific topic, which is studying the interplay between metabolomics and various aspects that we measure on human cohorts, in particular the microbiome. The very basic fundamental questions that we asked in this particular research was trying to establish what actually drives and what determines, which factors determine levels of different circulating metabolites in our blood. The reason why that is important, we know that, as Greg mentioned before, we know that molecules, the small molecules that circulate in our bloodstream, are going to be key to our health, and many of these molecules may be pathogenic and disease-causing. And so in many cases, when you identify one of these molecules as causing disease, what you'd like to do then is to modulate the levels of that molecule. And in order to do that, you have to understand what is actually responsible for generating its levels. And if you had that ability, like appears here on the slide at the bottom, then you'd be able to modify the component that determines it, be it diet or be it the microbiome, intervene in that component, and by that, modulate the level of the small molecule of interest in the blood, and then by that, have an impact on health. So that's why determining the factors that drive metabolites is very important. So this was the goal in this project, and in order to do that, we made use of a very unique cohort that we've been assembling in the past several years. This is part of a project that we called the Personalized Nutrition Project. This project had the aim of developing personalized nutrition approaches for people based on clinical data and the microbiome. What we did, and the original study was published in Cell in 2015, was to recruit 1,000 participants, connect each of them to a continuous glucose monitor, which profiled their blood glucose levels continuously throughout a week. During that week, participants logged all of their food intake. And what we observed is massive differences in terms of how people's blood sugar behaves, even when they eat exactly the same food. And that data really, on an unprecedented scale, showed that there is a great need to personalize our nutrition, at least as far as blood glucose levels go, which by themselves are key for obesity, diabetes, and many other diseases. So in order to identify the factors that determine those personalized responses, we collected a very massive data set that included medical background and food frequency questionnaires, genetics, microbiome, physical measurements, blood tests, and metabolomics, which we profiled using the platform of Metabolon. And in that study, we were able to combine all of that data and predict personalized blood glucose responses. But then in the work that I want to focus on today, we use that same data set to ask a different question, as I mentioned, to try and now understand not the continuous glucose monitoring data, but rather the data of the metabolomics, and use all of this other very deep phenotyping. As far as we are aware, the most deeply phenotyped cohort in terms of the aspects that I mentioned measured here, in order to see and compare these different determinants and how they compare in explaining different metabolites. And since we have them all in the same cohort, we can compare their relative contributions. The metabolomics, as I mentioned, is the platform of Metabolon. We obtained over 1,200 different metabolites that are measured. These are very abundant metabolites. Over 1,100 of them were detected in at least 50% of the cohort. They cover a wide range of different types of metabolites, which you can see here, many lipids, amino acids, xenobiotics, and interestingly, there's also hundreds of compounds that are unknown. They give a reproducible peak in the mass spectrometry, but currently the compound is unknown. But we can still study it from an informatics perspective and see what may determine levels of those metabolites as well. So as the most stringent way to test our ability to determine levels of metabolites, we asked whether we can actually predict them in individuals in which we never measured the metabolites. At least we never gave it to the model. Of course, we measure it, and then we compare the output of the model to the actual measurement to see how well we are doing. But this kind of out-of-sample testing is, in our view, one of the most stringent tests to convince ourselves that we actually learned something useful. So the way this would work is, let's say that you wanted to determine or predict the levels of caffeine in your bloodstream, which is one of the 1,200 metabolites that was measured, and you wanted to do it from dietary information. So you would take the matrix of dietary information, which you can see here with rows corresponding to participants and columns corresponding to different features of the diet that were profiled. Then you learn a mapping, you learn a machine learning algorithm that tries to learn the relationship between the diet and, in this case, levels of caffeine. We used boosting decision trees, but one could use different types of machine learning models. And once you derive a model, you take a subject whose level of caffeine you didn't give the model during training, and you use the model to predict that level, and you compare it to the actual measured level. And you repeat this process every time, such that in the end, all of the participants were at least once held out and not learned as part of the model, so you can predict them. And when we do this for caffeine, the model actually works very well. So from dietary data, we can predict levels of caffeine quite well, as you can see in the predictions that we achieve here. But caffeine is just one of the different metabolites, different 1,200 metabolites that we'd like to predict, and diet is just one of the inputs from which we would like to predict the metabolite. So overall, we learned for each metabolite from every different feature group used as input, we learned a model, and we did that exercise that I just went through in caffeine. So overall, this resulted in over 13,000 machine learning predictors, and we could evaluate how well they're doing. And very interestingly, we saw that for over 90% of the metabolites, so more accurately, 92% of the metabolites could actually be significantly predicted by any one of those different feature groups that I mentioned, like diet, microbiome, sex, time of day, various body measures, and so on and so forth. What was interesting is when we asked which components are driving the strongest prediction, dietary intake was number one, but then microbiome was number two, and each of these was responsible for predicting levels of hundreds of different metabolites. And interestingly, they were mostly complementary. So those metabolites that could be well explained by diet were not well explained by the microbiome, and vice versa. Another interesting question that we could ask is there some characteristic to those metabolites that are predicted well by diet and to those metabolites that are predicted well by microbiome, and similarly for the other feature groups? And what we found was very interesting. So for diet, we saw that there was an enrichment in the metabolites that it predicted well. There was an enrichment for xenobiotics, which may be expected from dietary intake. Interestingly, microbiome was the only feature group whose predictions, the metabolites that it predicted well, were enriched for those metabolites that I mentioned before that are unknown. And that's very interesting because those unknown compounds, they could be equally important for our health. We just haven't discovered what they are. And what we now are discovering is that what's responsible for determining levels of many of them is most likely the microbiome. So the next step that we asked is we wanted to go deeper into gaining insights into some mechanisms, and so go beyond just saying that diet and microbiome explain features, but rather see which features of the diet and which features of the microbiome, which bacteria are actually driving the prediction. So we used a feature attribution framework, which I don't have time to go into, but I encourage you to read in the link that's provided in the reference provided here. Basically, it breaks down the contribution of the different features that you use in the model to assessing their relative contribution to every prediction that you make. And so when we can construct they're called shaft values. We can construct a matrix like this, where on the x-axis we have the different features. In this case, these are dietary features. And on the y-axis, we have the different metabolites. And every time you see a color, it means that there was a significant contribution from the feature on the x-axis to contributing to explaining the feature, the metabolite on the y-axis. So as an example, if we go back to caffeine, this is a nice positive control and would be expected that the most dominant feature, contributing feature in the diet that contributed to predicting caffeine is self-reported levels of drinking coffee. Similarly, for another metabolite, stachyderine, which we also predicted well from the diet, this metabolite is a known biomarker of citrus fruit, and indeed, self-reported consumption of oranges was the most dominant feature contributing to this prediction. And similarly, a nice additional positive control is another metabolite here, which is a known derivative of fish, and also people reporting eating fish was the dominant feature driving these predictions. So these are nice positive control. Of course, we have hundreds of other metabolites where the predictions are much less obvious, but these give us confidence that those less obvious predictions are interesting to explore and understand and have a good chance of being correct. We can then do the same exercise for the microbiome, where much less is known about the specific bacteria driving each of these predictions, and this is the corresponding SHAP matrix, so the feature attribution matrix now for the microbiome. And if we go to individual metabolites now that were predicted well by the microbiome, first you can see these metabolites are really, the levels of the predictions are quite outstanding. I just remind you that what the exercise we did here is we developed a model based only on the microbiome and then applied it to held out individuals whose metabolomic data we never saw. And the model that learned the relationship between the microbiome and the metabolite was able to drive the predictions that you're seeing here, which basically means that if we knew the microbiome composition of these individuals, this is the accuracy with which we could tell what levels of these specific metabolites they have. And you can see on the top there's two unknown compounds, which I mentioned. The microbiome is enriched in generating good predictions for these metabolites. And what is colored here now are bacteria that were identified using the feature attribution framework as driving these predictions. And so what this means is that now these individual bacteria are likely those that are driving, maybe in this case, synthesizing the metabolites that you're seeing here. And the importance of that, again, would be that if one of these metabolites is one that we think from some external sources, there is a good reason why we'd like to modulate its levels because maybe it has some role in disease, then probably intervening with these bacteria, perhaps a targeted probiotics with this bacteria could actually modulate the level of this metabolite, and by that, then consequently impact health. So then finally, of course, when we learn from such observational data, strictly speaking, we can't really refer to causality, and that really needs to be established with further randomized control studies, which we've done as proof of concept for some of the predictions. So what you're seeing here are, in the case of one specific dietary component, this is for whole wheat bread, there were some metabolites that the model predicted at the top are metabolites that are negatively affected by consumption of whole wheat bread, namely they should go down after consumption of whole wheat bread, and conversely metabolites at the bottom positively associated. So these metabolites, according to the model, are increased by consumption of whole wheat bread. So what we did was a proof of concept small RCT where we recruited 20 participants. We gave each of them sourdough bread for 1 week. We measured their metabolites, again, using the Metabolon framework before and after the intervention. And indeed, what we could see is that metabolites that were predicted to increase following sourdough bread consumption, on average, these metabolites indeed increase after 1 week of intervention. And conversely, metabolites predicted to go down indeed went down after consumption of sourdough bread. And these predictions were actually quite specific. So if we gave the same 20 participants, this was a randomized crossover trial. So each participant then for another week ate white bread, and we did not see an effect on these metabolites after consumption of white bread. So these predictions were held up in clinical trial, indicating that at least in this case, the predictions actually had a causal impact. So with that, I'll just very briefly summarize what I mentioned. I told you about a very deeply cohort of human individuals in which we've coupled all of these different phenotypes with serum metabolomics. We were able to significantly predict over 92% of the measured metabolites from one of the different feature groups. Diet or biome each explained hundreds of different compounds that were measured. I tell you this, but we also have results in independent cohorts. And I showed you proof of concept demonstration of the causal effect of these. Of course, there's numerous different hypotheses now that were generated by this work on how microbiome, diet, and other factors impact metabolite levels that would be interesting to then further explore and potentially give us now means of intervening and modulating levels of these specific small molecules in the blood. So with that, I think I will stop here.
I'd like to thank again our panelists, Nikole and Eran, for really, really exciting talks, innovative talks, showing how you can use metabolomics and the power of metabolomics to deconvolute the role of microbes in different systems, both immune responses in chronic diseases and predictive models for understanding human health and wellness and how diet is affecting that in populations. So with that, I think we can open the discussion for a Q&A. So please feel free to enter your questions. I would like to start out initially by an initial discussion, and for both Eran and Nikole, given that microbial heterogeneity between human subjects is known to be very extensive, does this heterogeneity complicate efforts at identifying microbiome-based disease associations? And how do each of you think that metabolomics can assist in simplifying this task? Eran, I guess you went last. I think it makes sense for you maybe to lead off with that, and then I'll get Nicole's thoughts after that as well.
Yeah, that's a great topic for discussion. So yes, indeed, there's been many works showing that if you look at the individual bacteria that we each carry, they vary hugely between people, and this is for many different reasons because we live in different geographical places and we eat different things. But the way, and so if we had to study that heterogeneity, it's much greater than that which we have in the human genome. And so we'd really need huge populations in order to study that. But what people have also discovered is that if you look at the level of not the individual type of species, but you look at the functional level, you look at the metabolic pathways that are represented by the bacteria, then there's a lot more congruence between individuals in terms of the bacterial functions executed by the different bacteria. Even if two individuals have different species, they would converge in many cases on the different metabolic and functional pathways. And the output of those pathways would, in many cases, be small molecules and metabolites, which then could be taken up by epithelial cells and reach, as we just saw, systemic regulation and impact health. So I think a lot of the heterogeneity in bacteria actually eventually would converge on metabolites, and so by measuring the metabolomics, that would be one way in which we could circumvent the heterogeneity and also study this hub, which I think, Greg, you had in very nicely in your presentation, the metabolomic being kind of a hub that's key to understand, with the microbiome being one of the effectors.
Great. Thanks for that really great explanation. Nikole, do you have any thoughts on that, especially within the context of like chronic diseases and inflammatory diseases?
Right. I would ditto what Eran had to say in that the important take-home message here is that there is less heterogeneity at the functional level. I would add to that within disease, what we're seeing is also less heterogeneity than what's associated with healthy microbial compositions. So that too.
So you're talking about alpha diversity, things like that, the complexity of the the composition of the community?
It can be associated with it. And it's just that within a disease population, you see more standardization of the composition so that there can be more aspects that are alike during disease. And it's going back to the less heterogeneity and functionality, where metabolomics is going to play a role, is really helping us to, in large studies, recognize what is noise versus what is attributable to either disease process or response to a therapeutic. So both as, prognostic biomarkers or an ability to stratify patient populations on their ability to respond will be a part of how that plays into alleviating some of the heterogeneity.
And speaking of, you mentioned response and non-response, responders and non-response. Do you think the role of the microbiome in that? So for example, where there are inhaled therapeutics, typically inhaled steroids as a primary treatment, the way I understand it. What do you think the role is of microbial communities in regulating that response to responders, non-responders? Can they be identified and can they be stratified in that way, and the role of that, you think?
That is certainly the point of what we're trying to accomplish at Siolta Therapeutics, is to be able to really, and it's not just metabolomics, although this is one of the tools in our bucket to look at how we can stratify patient populations based on their response. And we absolutely believe that the gut microbiome is going to have a role in who would respond or not respond to any given concoction of bacterial species based on what their microbial background is. Which then really dictates their metabolic ability to respond.
Sure. Great, thank you. We have a question from the audience. This is for Eran. Do you expect metabolite levels that cannot be predicted well by diet or microbiome to be a result of the microbes acting on specific diet components? And can these interactions be evaluated?
So I think there's multiple reasons why some of the metabolites we can't explain with microbiome and diet, and some of these may be because some of them we know, for example, are genetically determined, and we actually have evidence for some of that, and some of them may be driven by other factors. You also have to remember, and this we actually are seeing is also improving with time, that our ability, microbiome, despite everything that we learned, it's still very early days. So we're still even mapping and identifying new bacterial species. And as we do that, we're able to extract more and more information from the same microbiome sequencing samples that we did even years ago. So in other words, our ability to analyze the microbiome is constantly improving. And that will also explain more and more metabolites. And similarly with diet is known to be actually hard to record. So as much as we try to do a good job in that, I don't have time to go into all the complexity, but it's very complex to very accurately record diet among people. So I think the fact that we can explain what we can explain in held out samples is what we can explain, we explain, and that definitely is a lower bound to what we will eventually be able to explain. And yes, in the models that we've done here, we purposely have been looking at each component separately in order to compare them. And it's very well possible, we haven't done that yet, that when we integrate multiple components together, some metabolites would be influenced by the interaction between different input groups that we use, say even between genetics and diet, or the microbiome and diet, as was mentioned, and those would also be able to explain a greater deal of the metabolites. So definitely.
Okay, thank you. And I think there's going to be a follow-up question to that as well. So question for Nikole from the audience. Regarding Siolta's LVP platform, driving precision medicine through stratification and iteration, where did you get the human data analyses?
So all of that work was done out of our co-founder, Dr. Susan Lynch's lab at the University of California, San Francisco. It's all published work that you can actually reference on that slide with the 3 publications. And she worked with collaborators in 2 different infant cohorts in which the early microbiome was studied. One was in collaboration with Christine Johnson out of Henry Ford. That's the WHEELS study. And they were looking at risk factors associated with the development of allergy and asthma. And she had access to cross-sectional samples across within hundreds of infants and children in that study. And then there was a second study that she collaborated with Michael Cabana at UCSF, and that is the TIP study, in which they were looking at infants that were across the first year of life that had been that were at high risk for developing allergy and asthma based on familial history, and then in a cohort treated with probiotics and a placebo in the other cohort. And then all of the actual data analysis was done through collaborators in Sue's lab with different data sets to provide the information. Was there a particular, I'm looking at the question now. So yeah, the data analyses, certainly for the predictive potential of the metabolites to which ones were impactful in distinguishing high risk and healthy was done out of Sue's lab and with collaborators on that study.
Great, thanks. Could you comment very quickly on, you showed the global footprint of what the cohorts are for asthma, Copenhagen, and kind of this international consortium. Is there any more been done in terms of the microbiome and metabolomics, in terms of deconvoluting that, being able to understand from these diverse areas what constellation of the pathogenic factors or even loss of protective factors there could be from the microbiome, given the broad range of the cohorts?
Right. With the international cohorts that we've seen, where we're seeing consistency is in westernized nations, so Canada, US, Denmark. Jeff Gordon has done a lot of work in some of the Asian countries in early life microbiome, and they've shown that there is a difference between the developing countries' microbiome, and they've done this through a method which they call aging the microbiome. And so based on the composition of the microbiome, they can predict the age of the infants, and they utilize this in malnourished children to see how the microbiome is being impacted. And with these studies and a lot of this data, what they've shown is they can actually compare the developing cohorts with those of Western nations, and they do show differences in that. Now, if you look at particular organisms that are of interest that are differentiating high risk and healthy cohorts in the Western populations, there is overlap with some of what you see in their work in malnutrition. And the idea that we believe is happening and what Sue's work has shown is that there's microbiome-driven immunometabolism going on very early in life, and that this has the ability to impact the immune system in different ways. And so I would imagine that is a mechanism of action that is consistent across developing microbiomes and in infants, but the actual drivers of those immunological responses and that specific metabolites may be different between those two sort of broad groups. in the developing and
Very interesting.
westernized. So
Thank you.
Sure.
Eran, back to you, kind of a follow-up to what you have been discussing. So you discussed how various patient data can be used to predict metabolite levels, but can you discuss the reverse approach now? Kind of which would be using metabolite levels and/or microbiome composition to predict disease state and/or other anthropomorphic factors like BMI?
Yeah, absolutely. So we've been doing that work as well. We actually have several papers even published on this. We've recently done this in a huge cohort of 15,000 metagenomic samples that we had, and actually relating even to what Nikole was discussing before. So this is not in infants, but in adults. We, for example, can predict the chronological age of individuals based only on microbiome data, also with quite high accuracy, and also in BMI and glycemic control features as well. So these type of predictions are also similarly helping us to raise hypotheses as to specific metabolites or gut bacteria that are also potentially involved in these different traits or disease states. Again, we always have to worry about correlation versus causation. So the fact that we find something being predictive of something else doesn't mean that it causes it. It could be a result of that, which is why after we raise these hypotheses, then we dive deeper into trying to study the nature of the associations that we found and try to distill which of them would be causative and which correlative, and that requires additional work in studies.
Great, thank you. And I think, given that we're kind of running up against time here, one final question that I'd like to hear from each of you again, and that is, do you believe that global metabolomics really provides an advantage over single or limited clinical biomarker studies? Since we started with Eran the first time in this dual way, but Nikole, if you could answer first and then finish up with Eran.
I think this is a great follow-up to what Eran just said, and I'm going to give you a very dissatisfying answer, which is that they're both really important. And so untargeted metabolomics gives us a broader profile of what's going on. It also allows us to identify things that we didn't know were playing a role. But of course, this always has to be followed up with validation and more targeted assays to really show the actual mechanism by which these metabolites might be impacting the system downstream. So I think they're both vital.
Eran, last thoughts?
Yeah, no, I think that was a very good point, and I definitely agree. I think once there's a well-founded marker that we established, in the community that's clinically relevant, we definitely want to measure it. We want to understand it, the mechanism driving it, how interventions that we do impact it. But at the same time, I think having, untargeted metabolomics is probably the best way to look at small molecules, which are key, like doing shotgun sequencing is the best way to look at the nucleic acid content, and it's not just bacteria mostly, but not just bacteria that we sequence in, say, in stool samples, and similarly proteomics or gene expression. So I think definitely this technology gives us a way to look very broadly at in probably the most comprehensive way for further discovery, corroboration, validation of other results that we have.
Wow, thank you so much, Nikole and Eran. These were fantastic talks and fantastic and dynamic discussion afterwards. I think these are exciting times for metabolism profiling. A lot of work has come out recently showing the power of this in deconvoluting the role of microbial function in health and wellness. And many papers have come out in this recently showing that these can be used to show alpha diversity scores, such as what Nathan Price just published recently. So we look forward to continuing to establish the role of bacterial metabolism health and wellness and expanding our platform to be able to provide coverage for this novel area. Thank you all for attending, and special thanks to the speakers for participating today. Thank you so much.
Thank you, Greg.
Thank you. Bye.